Good morning and a warm welcome to Bangkok. I apologize for not being able to attend in person and want to thank you, the organizers, for the invitation to share an update on I-ELCAP's work in this session. At I-ELCAP, we focus on advancing early cancer detection using metabolism-based liquid biopsy. My name is Volker Liebenberg, and I serve as the Chief Medical Officer at I-ELCAP, a Swedish diagnostics company with the mission to prevent cancer mortality. The company was founded back in 2017, and today we have an international team of 22 engineers, scientists, and healthcare professionals with an exceptional industry experience. We operate from our R&D and production facilities at the Karolinska Institute campus in Sweden, and from there we collaborate with over 30 clinical partners across Europe and North America. As said, I-ELCAP is pioneering metabolism-based liquid biopsies. This audience is well aware of the biological systems linking genotype and phenotype. Our early research in systems biology has identified glycosamimolglycans as the most deregulated biomolecules in cancer. GAGs, as they are usually abbreviated, are long unbranched repeating disaccharides units essential for connective tissue structure and cellular signaling. They play a key role in cell invasion, metastasis, and angiogenesis. Such metabolism-based biomarkers have strong potential. Nevertheless, they remain underexplored, mainly due to the analytical challenge which we're trying to solve. There are 17 different GAGs in the human body. Quantifying them allows to create what we call GAG profiles, or the GAGome. Based on them, we calculate GAGome scores via proprietary algorithms. These are numerical values ranging from 0 to 100, correlating with the predicted probability of a specific clinical event. For example, a cancer diagnosis or a recurrence detection. Over the past 5 years, we have developed high-quality distributable kits to enable precise testing, and we aim to distribute them to clinical laboratories. The workflow process starts from 100 microliter of either blood or urine samples. The GAGome kit and its instructions can be used to extract glycosamimolglycans, and then the actual measurement takes place on a commercially available mass spectrometer. Then the GAGome score is calculated with a cloud-based software and then dichotomized into a positive or a negative result determined by a cutoff. Such a workflow takes about 2 days to complete, and the first 2 products that we are aiming to launch are in renal cell cancer and in lung cancer risk prediction. Meanwhile, we have completed our first large-scale clinical studies towards clinical validation. In case of a cohort study, as shown here, there were 5,500 presumed healthy individuals, including 827 cancer patients, and we demonstrated that GAG changes can be detected up to 5 years before a clinical cancer diagnosis across multiple types of cancer. The diagram shows the absolute risk of developing cancer over time for several cancer types, analyzing urine at baseline. This GAGome score has been found to be independent of other known risk factors, for example, age, smoking, and in combination, this illustrates the potential of GAGome scores. Now, for the development for a specific indication, we need a lot more detail. As an illustration for this audience, I included plasma GAGome scores from a lung cancer study that has just been accepted for publication. These results here are from a retrospective cohort study, 50-50 patients with cancer and controls, about 1,300 patients, and we analyzed plasma samples at baseline. And what you can see here is the cancers versus the non-cancer controls, And the GAGOM scores demonstrate good discrimination between the lung cancers and all these non-cancerous conditions, which are typical comorbidities in this population. Now, the next step will be to confirm these findings in lung cancer screening cohorts. Let me close with an outlook of our development pipeline with the two indications to give you an idea about the timing. In kidney cancer recurrence monitoring, our leading project, we have scheduled clinical validation for the end of this year. So we will use samples from an international prospective observational study. And once this is successful, we expect another year until we can get this launched after regulatory approval. And in lung cancer risk stratification, we follow the same approach. This time, we aim to use biobank specimens from screening studies, first and foremost the Liverpool cohorts, but as well PLCO, UKLS, and probably 4 in the long run as well. And then if things go well, about a year later, we would hope to be able to put this on the market. This dual track development strategy uses biospecimens from prospective studies, which allows us to rapidly validate GAGOM testing. It will still take some years until we can expect clinical adoption. We consider this the start of our journey to improve patient outcome, and it will take a while. Nothing of this would be possible without the close academic collaborations, and we are very grateful for this and look forward to engaging more with this audience. Thank you for your attention. I look forward to your questions.